It would be remiss of me not to record. Okay, we're up and running. Sorry to interrupt. Here we go. I'll rewind this just a tad.
Bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system.
Just to stop, I'm going to be doing that quite a bit. This right here, that is a myofibril. It's sarcomeres just stacked end to end to end, okay? Keep that in mind. And then inside of these myofibrils, that's where the actin and myosin myofilaments, the contractile proteins are, okay? So, and this whole area is being bathed in sarcoplasm, okay? That's where the myofilaments live, inside the sarcoplasm.
Muscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance.
Just a quick review, all right? In the book, what are these structures here called? Remember? It's called the Z line. This video calls it the Z disc. Okay, that's the Z line. Yes, sir.
I think the book called it. I think I like the Learn Smart called it
Z disc. Yeah. So it interchanges it, but it's the same thing. Okay. You remember what this line is in the center of the sarcomere, where the myosin is anchored to. M line. That's right. The M line middle, and myosin. Myosin is anchored to it. Right. The Z discs are where the actin is anchored to, and it represents the ends of the sarcomere. Right.
The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin...
Sorry to interrupt again, but you notice here, all right, remember these little zones here that we talked about? This right here is the I band, okay? That's just where there's thin filaments only actin. So that's what gives your striated muscle that light appearance, right? And then where you see there's an overlap here, that's where the thick and thin, that's going to give the dark red appearance, right? And then you got the H zone here in the middle, and that is where the myosin occupies only, okay? So the I band, all right, and the H zone can completely disappear during maximal muscle contraction. You should be aware of that. You'll actually see it here, really small, and disappear.
To the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move.
We're going to talk about this
today.
This
action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a crossbridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds.
That's important to know. Because if you've read in the chapter about rigor mortis, your body's not producing ATP anymore. Your muscles get very stiff during rigor mortis, but that only lasts for a short period of time after death, because then the muscle tissue starts to break down, and those cross bridges disintegrate anyways. Right? But in order to release the myosin from the actin, you need ATP for that release. Okay?
Freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the crossbridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin.
You heard that. Two things that you need for a muscle contraction is calcium and ATP. You need the calcium. We learned this on Tuesday. You need calcium to remove the regulatory proteins off of the myosin binding sites on the actin. Okay? And then obviously we just saw what you need ATP for. Right? That's to release the cross bridge and to recycle the whole system to start another power stroke. Okay? So those are two things that you definitely need for contraction.
Exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, the muscle can produce enough force to move the body, allowing you to take note.
I want you to take notes, what she said. I know I cut her off. Just going back to this one diagram.
Travels down the T-tubules.
Okay. So just to kind of reinforce, all right, a couple concepts here so you understand. Right? This is the sarcoplasmic reticulum, this tube-like network, similar to the endoplasmic reticulum that we saw before. Right? At the ends of the sarcoplasmic reticulum, you have the terminal cisternae. That's this structure on either side of the T-tubule. Right? Calcium is found in the sarcoplasmic reticulum, also found in the terminal cisternae. You have what we call calcium pumps that are present in this structure. Okay, so after this muscle contraction, when you're done contracting, you have to scoop up, clean up all the calcium, and get it out of the sarcoplasm. We put it back here into our sarcoplasmic reticulum. So we have calcium ion pumps, which are going to put the calcium back into the sarcoplasmic reticulum. Right? Also, on the sarcoplasmic reticulum, we have the calcium-gated channels. Okay, remember when that action potential comes down here into the T-tubule, it locates what we call our voltage-sensitive calcium channels. They open up and they trigger the opening of the calcium channels. So calcium can then leave the sarcoplasmic reticulum and go into the sarcoplasm. Check it out.
And opens calcium stores. Calcium ion.
There you go. Okay? That's what happens. Right? So in a resting muscle, calcium is separate. It's not here, all right, in the sarcoplasm with the contractile proteins. It's kept hostage or it's kept sequestered here in the sarcoplasmic reticulum. Okay? So like I was saying, the calcium is the princess. This is its tower or castle, wherever it is, and it needs to be liberated. It needs to be freed. The action potential does that. Right? So keep that in mind. Okay. So we've seen that. So does this make a little bit more sense? Does this video make a little bit? And hopefully it'll make a little bit more sense after we finish today's, lecture. Right? Like I said, most people have a much better understanding of this, right, as we move along. All right, American Ninja Warrior and the Octopus Teacher. Okay, make sure, folks at home, that you answer the poll. You need to know. right? To be honest with you, really what I'm going to do when I get home is after I check emails and the attendances, I'm going to go play video games. Can't lie to you. Anyways, all right. Let's pull up the files here. Okay, so where we left off, we talked about the 3 steps or 3 phases, all right, as to what's going to happen here with, all right, a muscle contraction. All right, this is what leads up to our contraction. The first here is what occurs at the neuromuscular junction, right? The excitation of that skeletal muscle fiber. Remember, you're all pretty familiar with that, I'm assuming right now. The action potential, wait, let me find the picture. Much better. Okay, our action potential comes down, all right, the axon, hits the synaptic knob, triggers the opening of the voltage-gated calcium channels. Calcium is in high concentration outside the cell, thanks to our calcium ion pumps, right, that have pumped it out. Okay? So it diffuses, meaning it goes down its concentration gradient through the passive process, enters into the synaptic knob, coats the synaptic vesicles with its positive charge. It's drawn towards the plasma membrane over here by the synaptic cleft, releases the neurotransmitter through the process of exocytosis. Right? Now I can actually tell you what the neurotransmitter is. It's acetylcholine. Acetylcholine then diffuses across the synaptic cleft over to the motor end plate here, where it looks for its receptors. Okay? Finds the receptors on these channels here, opens up these channels, and sodium is able to enter in. Similar to what we saw when trying to develop an action potential in a neuron. Okay? 2 things. Our resting membrane potential for a muscle cell is -90, not -70. Okay? Neuron is -70. And the threshold value is -65, not -55. It's the same concept. We need to get to that threshold value. We have to change that resting membrane potential enough, okay, to trigger an action potential. So that's what we're starting here, okay? So we're starting to trigger what we call an EPP, an end plate potential, okay, which is similar, not the same, but similar to a graded potential. They're small, they're local, right? They will die out, okay? Not like an action potential, which is an all or nothing thing. Once you get an action potential, it's gone. Right? So that's the first step. The next step is this one right here, our excitation-contraction coupling. Now we need to generate, right, enough of a difference in that resting membrane potential to cause what we call muscular action potential. And then that muscular action potential needs to find a T-tubule somewhere. Okay? So when an action potential occurs, when an end plate potential occurs, it's literally the same thing as throwing a big rock into a pond. It goes out, just spreads out. And that action potential shoots out from the source, from the origin source, and it looks for T-tubules. Okay? And then it's going to go down those T-tubules, right? Like a well. Think of the T-tubule as a well. As that action potential goes down into the T-tubule, it's going to look for the voltage-sensitive calcium channels to trigger the release of calcium. Okay, because remember, you cannot have a muscle contraction as long as the troponin and the tropomyosin regulatory proteins are sitting on the myosin binding site of actin. Okay, so that's what we're seeing here. Now, a little bit. Okay. We've now generated our muscular action potential, same situation, sequential opening of sodium and potassium ion gates, okay, or excuse me, voltage gates, right? They open up just like we learned in the neuron, right? And the action potential moves along parallel to the sarcolemma, that's the plasma membrane, okay? Finds a T-tubule, goes down into the T-tubule until it makes connections or contact with a voltage-sensitive calcium channel. Then it causes that voltage-sensitive calcium channel to trigger the opening of these calcium channels. And these calcium channels, which are located on the terminal cisterna here and on our sarcoplasmic reticulum, they open up and they let calcium that's trapped inside leave and go out into the cytoplasm, or the sarcoplasm. In the sarcoplasm, that's where our myofilaments are. the contractile protein, okay? The myosin and the actin, okay? That's what we're going to pick up with, which was right here. Okay, sarcomere, crossbridge cycling. This is the next step, okay? So this is the final phase, right, of our generation of a muscle contraction. So here's what happens, right? And I've told you the story, you just saw it on the video. Calcium, right, diffuses into the sarcoplasm, right? It binds to the troponin, which sits on top of that rope-like regulatory protein, that tropomyosin, right? It causes it to shift off of the myosin binding sites, right, on the actin. So now that myosin binding site is exposed. So we can start to begin our cross-bridge cycling. Right? See that? Okay, so our calcium comes in, just like in the video, sits on this little cup-like structure on the troponin, right? And it causes the tropomyosin to shift off the binding sites here, the myosin binding sites. So now our myosin globular heads can connect to the actin. All right? A couple things about the calcium binding. Right? One, you need to know that calcium binds onto troponin, not tropomyosin, but troponin. Right? It causes this conformational state changes. It changes the shape of the regulatory proteins, right? And it causes the troponin-tropomyosin complex to shift away from the myosin binding sites. Now they're exposed. So myosin can now bind onto the actin, so we can get our cross bridge. Okay? Not too bad? A little bit, you know, I know there's some memorization involved. Let's check on our poll. I got one person that hasn't responded at home. It's a tie between the octopus teacher and then the American Ninja Warrior. Ah, you missed it. It's I'll tell you about it. Octopus teacher. Oh no, wait, it's still tied. No worries. It's a documentary on Netflix. Right. So there's 4 steps to the cross-bridge cycling. All right. So what we're going to do is, you know me by now. I'm going to do one step, show you a picture, then go to the second step, show you the picture for that. Okay. So we'll break it down nice and easy. Okay. So we're ready to begin. All right. Our cross-bridge mechanism. Okay. So the first step, and again, these are repeating steps. So as long as that muscle is being stimulated by the motor neuron, and as long as we have ATP, and as long as we have calcium, we can continue this process here. Okay, so the first one is to make a cross bridge. Okay, so in this first step, myosin is in the cocked position. Okay, so what it does is it extends across that gap there, and it forms that cross bridge between the myosin and the actin. Real easy. I'm going to come back to this slide. All right, so I'll show you this first step here. Okay. All right. So there's an ATP here, right? And what happens is, right, it phosphorylates, right, and it turns into the inorganic phosphate and ADP, right? But this is what causes that head to cock, okay, in the cock position now. Oh, I'm in the wrong one, y'all. Sorry. Where am I supposed to? Sorry. There we go. Okay. Whew. It's not working. All right. So here we are in the first step. Okay. So we have our cross bridge formation. So the myosin head is in the cock position. Right. What it does is simple. It's just going to extend across that gap because we now have the actin or the myosin binding site exposed, and it's going to connect to that myosin binding site. Okay. Simple. Right. That's step 1. Right. So step 2, right, is the power stroke. This is when we start the contraction. Power stroke is when we start to create movements. Right. So this is where the myosin head is going to now swivel and kind of pull the actin, all right, past the myosin, or it's going to pull the thin filament past, all right, a specified distance past the thick filament. Okay, as it does that, all right. So just think of it as it's moving the actin past, it releases the ADP and the inorganic phosphate. So now we have, remember, because myosin has two binding sites. One's for the actin, the other one's for ATP. So now that ATP binding site's available because the ADP and the inorganic phosphate flew off of it. But at this point, okay, with the power stroke, the myosin head is still attached to the actin. It's still stuck there. Okay? I'm going to come right back to the slide here. All right, so that's what we're seeing here. All right, so here it was when it connected, and now it swivels, it does its power stroke maneuver, and as it does that, all right, it brings the thin filament, moves it by it. Okay? And at the same time as it's doing that, our ADP and our inorganic phosphate pop off. So we have, all right, the ATP binding site is now available. Okay? Remember what I said, we need ATP to release that crossbridge formation. Okay, so that's an energy requiring component to this. All right, let me go back to the second step, just in case you need to write some more stuff down. Okay, so, okay, the power stroke, the myosin head swivels, right? It moves the thin filament past the thick filament, and as it does that, all right, our ADP and the inorganic phosphate are released, thus exposing that ATP binding site. Ready for another ATP to bind on, all right, to our myosin head. Okay? Well, that happens here, okay, in our third step. Okay? That's when we're going to release the myosin head. Okay? Because either we're going to stop the contraction altogether, all right, or we need to reset everything for more contraction, okay? So during this phase, our ATP, our new ATP, binds on to the ATP binding site, and that causes the release of myosin from the actin, okay? This doesn't happen in rigor mortis. Okay, so I'll come back to this right here. Let's look at the third picture. Okay. All right. So at this point, we're still connected, right, until ATP, right, binds onto the ATP binding site. When that happens, then the myosin head is released from the thin filament. Okay. Pops off. Okay. For either relaxation, for the contraction to end, or for another power stroke. Okay. That's our two options. We can stop doing the contraction, or we can reset the myosin head for another power stroke. All right, so phase 4, or step 4, excuse me, okay, is us resetting that myosin head. Okay, so we need to reset the myosin head for another power stroke. All right, so as we do that, okay, we hydrolyze the ATP. That means that we break it down. All right. We break it down into that ADP and that inorganic phosphate. Now you need to know this enzyme. I don't think I told you guys this. I told the other section this. Anytime you see A, S, E on the end of a word, okay, that's an enzyme. Acetylcholine esterase is the enzyme that breaks down acetylcholine into acetate and choline. Right? ATPase is the enzyme that breaks ATP down into ADP, inorganic phosphate. Okay? So, and this we'll talk more about this later on, because this can actually occur quickly, for quick muscle contractions, or it can happen slower. All right, so that plays a role. We'll talk about it maybe today. If not today, the next class. Okay? So this enzyme, all right, splits the ATP into ADP and inorganic phosphate, and that gives us the energy to reset the head, or what we call cock the head. Okay, so looking here you can see, right, where we once had ATP attached, right, ATPase, right, is going to break it down into ADP and inorganic phosphate, which are still attached to the myosin head, all right, but it puts it into the cocked position. So it's ready for another power stroke if we're going to do a contraction, right, or we just relax and not do a darn thing, okay? So this whole cycle, what you're seeing here, right, is a repeating cycle, right, as long as we have action potentials coming from the motor neuron, as long as we have calcium and ATP available, right, then this can repeat, right, as long as that happens, right? Make sure everybody got this written down. Crossbridge cycling is not terribly detailed. Not too bad. Just go ahead.
It kind of reminds me of the things like almost like talking about a revolver. So you have that little
Yeah.
guy there. It reminds me kind of doing that kind of motion.
Yep. Very similar. Very similar. Okay. So these 4 steps are illustrated here. So what I would suggest is, again, I'm a picture person. I love illustrations. Go with this. All right. Figure 1013. You understand all this in writing. You can see the pictures. It'll explain. But that video does a good job too. You know, it's 4 minutes and 25 seconds. That's why I love it. It's jam-packed with awesome information. Okay? Okay, so going back and just telling you what I just said here, all right? We will continue this whole crossbridge cycling as long as we have calcium and ATP present. And obviously, as long as, it goes without saying, but as long as the motor neuron is still sending action potentials too, to stimulate this whole process to occur. Okay? So this causes, right, the contraction of the sarcomere, which means our Z discs move closer together. Right? The H zone and the I band will either get very narrow or pretty much disappear altogether. Okay? But understand, the thick and the thin filaments, their length never changes. They do not change. They just slide by each other. Like my fingers. I take my fingers and I'm doing this motion here. My finger length isn't changing. All right? They're just sliding by each other. The distance in between, right, my wrists gets shorter because I'm just sliding my fingers by each other. Same type of concept. Right? Those myofilaments never change length. Okay? Just the distance between the Z discs gets shorter. That make sense? Kind of, sort of? All right. So this next picture here kind of illustrates that. All right. You can see here, right, we've got our relaxed muscle, the biceps brachii. So you can see here's the normal length, all right, of a resting sarcomere. Right. So we can see the I band, all right, and the H zone. All right, it's all good. But now when we start to contract the muscle, you can see that the length of the thick and thin filaments doesn't change. Right? It's just the amount of overlap that changes. That will increase, okay? So that H zone will disappear, well, it'll get smaller or completely disappear if it's a maximal muscle contraction. And the I band is smaller or it can disappear during maximal muscle contraction, okay? So just keep those qualities in mind there, right? So what is the function of calcium in skeletal muscle contraction? I like to think that everybody's pretty good with that, right? We need our calcium, all right, to remove the regulatory proteins, right, the troponin tropomyosin complex, off of the myosin binding sites. So we can expose the myosin binding sites, right, to myosin, so we can now create our cross-bridge cycling. Okay? You guys feel pretty good about that? All right, good. All right, what about this one? What causes the release of the myosin head from actin? ATP. Okay, it's an energy requiring process, so ATP, when it binds onto that, is going to release the myosin head from the actin. And then what resets it is when we involve an enzyme, ATPase. Right? ATPase is going to split ATP into ADP and inorganic phosphate. Okay, that's what the P and the I stand for, the little I, inorganic. Okay. So keep in mind, you need to know that. I've seen this question here, all right, this 2-part question has been asked on tests. That's an easy test answer for you guys to get correct. Okay? I hate for you to miss that. All right. So, you know the saying, what goes up must come down? So when you contract the muscle, you have to relax the muscle, right? So a lot of what involves muscle relaxation is kind of like it's not exactly, but it's somewhat like the opposite of the muscle contraction, okay? So let's take a look at it. It's a big slide here, so bear with me, okay? But if you think about it, and I'll go through it step by step, it makes sense though. All right, the first one here, stopping the action potential, okay? If we stop the action potential from the motor neuron, right, we're not going to release acetylcholine anymore from the synaptic knob, all right? So therefore, we'll never have the excitation-contraction coupling, right? And we'll never have crossbridge cycling. So that's a good way to relax a muscle, right? Another way is to, if there's acetylcholine, all right, that's lingering in the synaptic cleft. Remember, we talked about 3 different ways to clear a neurotransmitter from the synaptic cleft, okay? There's reuptake, where you have these pumps that will just pump the acetylcholine back into the synaptic knob. The glial cells will go in and act like a hoover and suck it up. And the third one is recycling. We break down that neurotransmitter. In this case, we use acetylcholine esterase to break down the acetylcholine into acetate and choline, okay? So we do that. That process is called hydrolysis, and it busts down acetylcholine into its building blocks, okay? So by doing that, it causes the acetylcholine receptor, if there's no neurotransmitter there, right, those gates close. So sodium and potassium don't cross the plasma membrane. If sodium and potassium don't cross the plasma membrane, we never reach our threshold value of negative 65 millivolts. So guess what? No action potential. Okay? And if there's no action potential, then that means, all right, those calcium channels that are located, all right, on the terminal cisterna and on the sarcoplasmic reticulum that are and are voltage sensitive calcium channel, all right, those close. So calcium now is no longer getting released from the sarcoplasmic reticulum in the terminal cisterna, and at the same time, our calcium ion pumps is pumping calcium back into the sarcoplasmic reticulum. So calcium returns to the sarcoplasmic reticulum through those pumps. And since calcium's not available, guess what? Our regulatory proteins, troponin and tropomyosin, slam close, I shouldn't say close, they slam back over the myosin binding sites on actin. Well, since now we've blocked, right, myosin binding sites, myosin cannot cross-bridge with actin, so our muscle then returns back to its original shape, okay, due to elasticity. Right? And one of the proteins that's responsible for elasticity, you guys remember? There was 2 proteins, I only mentioned them briefly on one slide. One has a condition named after it, muscular dystrophy. There's dystrophin, okay? But that's not the protein I'm talking about. The other one, it's called connectin. On that one diagram, it shows it looks like a spring. Okay? And it's the connectin that helps the muscle with its elasticity attribute or characteristic. Okay? So if you read through the slide, it's almost like a step-by-step kind of process here. Okay? You know, I know that you know this. It's almost like a kind of a step. It's like a story. Okay? That's why I like this chapter, because it's a story-like chapter. All right. So this question asks, how do acetylcholine esterase, so you see the last 3 letters, esterase, that's an enzyme, and calcium pumps function in the relaxation of a muscle? Right, well, we know that acetylcholine esterase breaks acetylcholine down. So it stops acetylcholine, all right, from performing its function in the synaptic cleft. Okay? And then our calcium ion pumps will continuously pump calcium from the sarcoplasm back into the sarcoplasmic reticulum. It removes it from the sarcoplasm. And what lives in the sarcoplasm? Actin and myosin. You know, I kind of it just jumped into my head, and maybe it was because I was watching The Octopus Teacher. I don't know. But I kind of think of like troponin and tropomyosin. You ever seen sharks swimming in the ocean and they got those smaller fish like hooked onto them like lampreys? That's what I think of the regulatory proteins, troponin and tropomyosin. They're like the lampreys on the shark, right? So in this case, they just slam back over the myosin binding sites that prevents the cross-bridge cycling there. All right, so I briefly talked about energy production last class in muscles, okay? As you know, the energy currency in the cell is ATP, right? So on site, on hand, right, your muscle cells have enough ATP, all right, for a 5-second contraction or 5-second amount of maximal exertion. That's not long, okay? So we have to figure out some additional ways to produce energy for muscle contraction, okay? This first one here, okay, if you look at this term here, myokinase, myo, right, muscle, kinase is an enzyme that likes to put phosphate onto things, okay? Likes to just stick phosphate on stuff, okay? So your muscle cells have this enzyme available, okay? And so it likes to put phosphate onto ADP, because what happens when you add a phosphate to ADP? What do you get? ATP, that's right, okay? So this enzyme hangs out in muscle cells, and it goes around, and because what happens, we just saw what happens during the cross-bridge cycling, right? You saw what happens to ATP. What happens during, all right, the actual power stroke? Well, what happens to ATP? It attaches onto the myosin head, right? And then what happens to it? It breaks down, right? ATPase breaks it down into ADP, inorganic phosphate. So you have this ADP that's just floating around in your cytoplasm, in the sarcoplasm. So it makes sense to have this enzyme there to fix it back up. Okay, we're going to build it back up. We're going to build it back up to ATP. We want to make another ATP for another muscle contraction, right? So there's 3 ways, okay, to generate ATP in skeletal muscle. And we talked about this earlier, not today. We talked about it last class. Remember aerobic cellular respiration, mitochondria, right? And you had that myoglobin, that awesome molecule that stores oxygen inside your muscle cells, so you have oxygen on hand, so you can have cellular respiration. Where oxygen helps to get, all right, pyruvate. You don't need to really know this for this class, but pyruvate is what we call an intermediate product. We break down glucose, we break it down into pyruvate, and then we take the pyruvate and we bring it into the mitochondria. And the mitochondria breaks it down more. It makes ATP. Glucose, 1 molecule of glucose. If you have no oxygen present, you can only get 2 ATP from 1 molecule of glucose. If oxygen is present, okay, then you can get 32 ATP. So it's well worth it to have oxygen on hand. Okay? Well, anyways, when we're breaking down glucose, somewhere in that breakdown process, right, you get pyruvate. And so pyruvate, then if oxygen is present, gets brought into the mitochondria, and then it will get broken down so we can get more ATP. All right, so the first way we're going to talk about getting ATP is from creatine phosphate. The key term here is phosphate. All right, let's talk about it. So this molecule has, all right, a very strong bond, or well, I shouldn't say strong bond, but a high energy bond, all right, between the creatine and the phosphate here. All right, so what we do, and I'm going to come right back to this slide here. I like pictures. I like teaching pictures. All right, so let me do this side first. Okay, this is what happens in a normal muscle cell. Okay. We're having contractions. As we're breaking down ATP, we got a bunch of ADP kind of floating around. We need to recycle this ADP or convert it back into ATP. So your muscle, all right, your muscle cell has myokinase. remember what I said, kinase is an enzyme that likes to put phosphate onto things. So this is what will happen, right? You got two ADP molecules, okay? So my myokinase comes in, it's going to pluck one phosphate off of one of the ADP molecules, and it's going to stick it onto the other. So then what happens after that, we're left with one ATP molecule, which is good, that's great, but then we're stuck with one AMP, okay? That's adenosine monophosphate, means it only has one phosphate attached to it. T is di, 2-phosphate, then T, obviously you know, okay? So that's what happens, and so that's great. We have this other component here, okay? Creatine phosphate, that's what the CP stands for, okay? So in this situation, you've got one molecule of ADP floating around, and then you've got creatine phosphate here, okay? You have this enzyme called creatine kinase, but the important part is kinase. So this enzyme is going to pluck the phosphate here, okay, off of this molecule, and it's going to stick it onto this molecule, giving us ATP and leaving us with creatine. So now you know why, and you might be interested in this, why bodybuilders and people that work out take creatine. And they'll take this creatine phosphate before they work out or during their workout because they're supplying their body with a ton of, this. Okay? So this enzyme can do its thing. So they can get more energy during their workouts. Okay? So that's what we see. We see, all right, this enzyme, all right, this creatine kinase, right, which is going to just transfer the phosphate from from creatine phosphate onto ADP, which will give us ATP. So this is nice. This will give us, it might not sound like a lot, but it gives us additional 10 to 15 seconds of additional energy. Okay? Does that kind of make sense? All right, so as long as you know that, kinase is an enzyme that likes to put phosphate onto things. We've got the myokinase at work all the time, okay? And then we've got our creatine kinase. All right, so the second, all right, way that we can generate ATP in the body is glycolysis. Okay? Glycolysis is essentially the breaking down of a glucose molecule. Right? It does not require oxygen, so therefore it is what we call anaerobic. Okay? The long story short is glucose gets converted into two pyruvate molecules. Right? So we can get glucose from the blood, our blood sugar, or it's stored within two organs in our body. What are the two organs in which we store glucose in? Muscles and liver. That's right, muscles and liver. So when you're sleeping, you go to bed at a normal time, around 3, nah, that's actually too soon. Around 4 or 5 in the morning, your liver is breaking down the glycogen that is stored there. Right? Because you burnt through a lot of your glucose stores in your immediate cells. And so the liver starts to go into a glycogenolysis and it starts to break the glycogen down so it can keep your blood sugar levels at a decent rate. Okay? So that's where we're going to find our glucose. It's either going to be found in the blood or it's going to be in a stored form in glycogen in your skeletal muscles or in your liver. Okay? So if just through the process of the glycolysis, we're only going to make 2 ATP. It's not a lot, but it's better than nothing. Okay? Something you need to know is this occurs in cytoplasm, the cytosol. That's why we have to bring glucose into a cell. All right? We get to talk about that in 211. All right, so the last form, or the last method, right, of us being able to produce energy for a cell is, and we've talked about this one, aerobic cellular respiration. Okay? Aerobic means with oxygen. Okay? It deals with the mitochondria. Okay? So we always will start off with glycolysis, and then at the end of glycolysis, we end up with 2 pyruvate molecules, okay? We have to get the pyruvate as long as oxygen is ready, or available, all right? It will allow us to take the pyruvate and bring it into the mitochondria, okay? I won't get into all this, but that pyruvate is oxidized to carbon dioxide. That gets into the Krebs cycle, the electron transport chain, and you'll get to that in 211. But what I do want you to understand, right, is, right, from those 2 pyruvate molecules, we'll generate 30 ATP. Okay? As we break down those pyruvate molecules, right, and we create several different intermediaries, okay? As we do that, we will generate energy, and we transfer these energy bonds into these 2 structures here, NADH and FADH2. Right? And then it's like going to the casino, gambling, right? You win a bunch of money, but you don't get it in actual currency, you get it in chips, right? So you're going to take those chips at the end of the night, and hopefully you're a big winner. You're going to take those chips at the end of the night, and you're going to go cash them out. And that's what the electron transport chain is. These are your chips, okay? You're going to take them to what we call the electron transport chain. You're going to cash all those chips out. At the end of the night, you'll wind up with 30 ATP, okay? So the nice thing about this process is, right, you get a lot of ATP. So even better is if you find a source of fuel that is what we refer to as energy rich. Well, we talked about that when we talked about the different types of macromolecules. Remember when we were talking about carbohydrates and proteins and nucleic acids and finally lipids and fat, okay? And one of the functions of fat, we talked about insulation, talked about cushioning, but remember energy, okay? Energy, our triglycerides are an amazing source of energy because a triglyceride has a glycerol group. More importantly, it has 3 fatty acids attached to that triglycerol group. That glycerol group, right? And those fatty acids are a long chain, okay, of these molecules here. Okay, carbon is going to be the backbone of this molecule. What happens is, right, you undergo, right, the breakdown, and you'll talk about this again in 211, you break down of the, it's called beta oxidation. You'll break down these fatty acids and you just start chopping off these carbon units. Boom, boom, boom. And as you chop them off, you generate fuel, which will give you ATP. Okay? So the longer the fatty acid chain, the more fuel you'll get. So believe it or not, your skeletal muscles love fat. Your body does. It's a great energy source. That's why your body stores it. You know, it's a I want to say instinctual thing. It's a survival mechanism that over hundreds and thousands of years that our bodies have actually come to this present day situation in which it prefers fat because it's an energy rich source for fuel. All right, so that's why when I talk about triglycerides, it truly is the preferential fuel, right, for your body. Yep. Because, and again, you'll talk about that a little bit next semester too, when you're talking about the formation of certain lipids like the VLDLs, LDLs, and HDLs, and different types of cholesterol. Because if the levels are high, then you're more prone to generating atherosclerosis is a possibility. But there's other variables that can affect that. Inflammation, certain conditions. But yeah. Exactly. Most of the time, though, when you get atherosclerosis, it's usually due to an actual physical insult to the blood vessel first. So some sort of inflammation has to occur. It just doesn't happen on its own. Because naturally, your blood vessels are slippery, okay? And actually, they repel, the walls usually repel structures because they produce certain chemicals that don't allow stuff to stick onto them. But when they suffer some sort of physical injury or damage, and inflammation is usually one of the causes there, then it loses that attribute, and then it makes it prone for stuff to stick on. If it doesn't resolve itself, then it starts to build up that plaque that you're thinking about. Yeah. Okay, so what this is showing you here, all right, again, I'm not going to go into this. This is a great figure, 1017. This is pretty much what I just talked about, and it just kind of gives you the different diagrams that you can kind of go from. Okay. All right. All right. So, kind of put into perspective when your body is going to use what. Okay. If you are going to do short bursts of energy, stuff that lasts 10 seconds or less, right, like a 50-meter sprint, you're primarily going to use that phosphate transfer system, the myokinase and the creatine kinase. Okay. That's what you'll primarily use. You're going to do something that's going to be pretty demanding on your body for about 1 minute. Examples of 400-meter sprint. Okay. Then you're going to use, again, all right, the phosphate transfer system. That's only good for about 10 to 15 seconds. Then you'll start to use glycolysis after that. Okay. Now, if you're going to run a marathon, or if you're going to do like a mile run or the 15-meter run, something that's going to last longer than a minute, you're going to use all three of those systems, right? Primarily aerobic cellular respiration, as long as oxygen is available. But I tell folks an easy, I shouldn't say an easy way, but a good indicator that if you're running, okay, or working out or jogging, or as like they call it over in Europe, jogging, all right? If you're doing that, all right, you ever saw the movie? Do you know what I'm talking about? It's a San Diego. San Diegans. Anyways, so if you're able to talk while you are working out, when I say working out, like running, okay, if you're able to talk, you are exercising aerobically. Right? In fact, when I was in college, I used to go running about 3 miles every night with a friend of mine, and we would talk the whole time. We were like two middle school girls gabbing about whatever, but we didn't run at a speedy pace, but it was definitely faster than jogging and slower than sprinting. But I tell you what, I know that I was burning up tons of calories, tons and tons of calories. We talked the whole time. Okay. Yeah, that was part. Yeah. Oh, yeah. It's true. Yeah. Well, when I was at the Citadel, we had to sing. We called them Jodies, and you had to sing the whole time. And now I used to drive a Cadillac. Now I carry it on my back. I could go on and on. All right. Anyways, additional ATP is made immediately available in muscle tissue through which unique phosphate-containing molecule? That's this guy right here. It's in phosphate. Okay. And then it's the CK, the creatine kinase, that is going to pluck the phosphate off the creatine and stick it onto our ADP. What are the various means for making ATP available in a 15-meter race? All three of those processes. Okay? Primarily our aerobic cellular respiration. Then our anaerobic cellular respiration is the glycolysis. I'll just abbreviate it here. And then the phosphagen system. All right, the myokinase, the creatine kinase. Okay. All right, I love this next, and it's only one slide. It's not very long. All right, I love this, oxygen debt. All right, it's a great concept to know. All right, because it's very important, especially if you're going to be exercising or working out and you want to continue to do it. Or say you're like, all right, I want to change my workout and do a different type of workout. You need to restore the amount of oxygen that is in your cells. Right? Now you have two molecules in your body that like to carry oxygen. Hemoglobin in your blood and then myoglobin in the muscles there. Okay? So this oxygen debt is basically how much oxygen that you need, right, after you're done working out, all right, to get your body back to how it was before you started working out. Okay? And during a workout, and it's really important, depending on what type of exercise you're doing, right, that you're utilizing your rest time. I know that some folks, their rest time in between repetitions depending, they should use it wisely, mainly because of oxygen demand. We want you to restore your oxygen levels to how they were, get them as close to what they were before you started working out, right? But ideally, it's so we can replace the oxygen on our hemoglobin and myoglobin. Right? Also, during this time, your body will make it a point, right, to insulin gets released, and this is why exercise is so important for people that are diabetic, right? Because insulin will then take, right, any glucose that's circulating into your blood, and it will start to put it into the tissues, the cells, to replenish, all right, their glucose levels or their glycogen levels, because those cells will store, all right, that glucose as glycogen. Okay. Then at the same time, we're going to take all that extra ADP that's floating around in the cytosol and convert it back into ATP. All right. Same with the creatine phosphate. We'll replenish that. Lactic acid, all right, is a byproduct that occurs during these metabolic processes. In fact, I didn't go over that slide here, just real quick. Lactate formation. Okay. So this is what happens when we have very little oxygen. Okay? What will happen is our pyruvate, okay, will convert into lactate. All right? And it's not ideal. Most people are familiar with lactic acid because sore muscles, and that's what happens. A lot of times you'll get a huge buildup of lactic acid in the muscles, and it causes like a cramping and painful soreness in the muscles, right? But also, right, our lactate can actually be converted back into pyruvate and used for energy later on. Okay, so your skeletal muscle can use it as fuel. All right. Also, cardiac muscle, right, will use it if it has to. It's not ideal. Okay? Lactic acid or lactate is not an ideal fuel source. Right. But your body makes do. You'd be surprised what your body does when it has garbage to deal with. Okay, so you'll learn more about the lactic acid cycle in 211. All right, but basically this occurs in the liver. Right, through this process here. I love gluconeogenesis. Gluconeogenesis is when we make glucose from non-carbohydrate sources. So fatty acids, we can actually convert. We can make sugar, glucose, from fatty acids. Lactate is another one of them. All right, pyruvate. It's really kind of a cool process. Okay. All right. Oxygen debt. Yes, muscle fibers. All right. So let's get into the different types of muscle fibers that are present in your body. Okay. So first, before we discuss them, I want to discuss the classification. How do we classify, right, these muscle fibers? One is the type of contraction. Is it quick? Is it slow? Okay, fast twitch, slow twitch. Okay. The second is how we're able to supply ATP to the muscle fiber. Okay. All right. So when we're going to classify, all right, the muscle fiber based on the type of contraction, all right, these three, right, categories help us to determine the type of contraction based on power, right, speed, and duration. Okay, overall power is the amount of force that we can generate. Obviously, speed and duration are self-explanatory. Okay? So, basically, the bigger the muscle, the more power. Think about it. Your quadriceps muscle group can generate a heck of a lot more power, all right, than your zygomaticus minor, okay, or your frontalis muscle, all right? So the bigger the muscle, the larger the diameter, all right, the more powerful the contraction, okay? Your quadriceps muscles have much larger muscle fibers, okay? And as you recall, the muscle fibers are those cells there, right? So the speed and duration, this is based on, and this is where I mentioned this earlier, and I'm bringing us back to this enzyme here, the ATPase, okay? This comes back to the type of that myosin ATPase, all right? And how quickly, right, it's able to break down ATP. Okay? Also, it also is dependent on how fast we can propagate an action potential along the muscle fiber itself, and how fast we can release calcium, right, from the sarcoplasmic reticulum, and also how fast we can reuptake that calcium. So there's a lot of factors involved in the speed and duration when we're discussing the type of contraction. Okay? Power is easy. The bigger the muscle, okay, the thicker it is, the larger the diameter, right, the more power can be generated. Right? Speed and duration, a little bit more involved in that. Okay? We're dealing with the type of enzyme, all right, that ATP enzyme on the myosin head. Okay? Also how fast we can propagate and promote our action potential, and then how quickly we can release calcium from the sarcoplasmic reticulum, but how quickly we can hoover it back up and suck it back up into the sarcoplasmic reticulum. Okay? All right. So let's get into the specifics here. All right. So when we're talking about our fiber types, we've got our fast twitch and slow twitch. I'm sure some of you have heard of these two before, okay? Especially if you've undergone any type of I don't even know if they talk about it in gym class anymore. All right. But our fast twitch, okay, this one here is going to have what we refer to as the fast variant of the myosin ATPase. Right? So this is how quickly we can break down that ATP. All right. Also, all right, these types of contractions for these muscles will be shorter. The length of time that they last for will be shorter. Right? But they will produce a relatively strong, forceful contraction. So for the most part, here's what you want to think about for fast twitch muscle fibers. Okay? More power, quicker contraction. And I mean that's self-explanatory and fast in the title fast. Okay? It's going to be a much quicker contraction. But you want to know why, right? We're going to be able to move our action potential at a much faster rate along the surface of the sarcolemma. Okay? And then how quickly we're able to, like I said, release calcium from the sarcoplasmic reticulum, and then how quickly calcium ion pumps can pump it right back into the sarcoplasmic reticulum following a contraction, okay, or stimulation, I should say. Slow twitch is easy, right? It just has the slow variant of the myosin ATPase. But slow twitch are great for endurance muscles. For example, your postural muscles. Okay, they're contracting right now as you're sitting there. You're not fatiguing because you're not falling over and slumping over. Okay. All right. So that is the type when we're talking about the type of contraction. The other classification is going to be how we supply ATP. All right. So there's two types here. Okay. You have oxidative and glycolytic. Now you've seen glycogen, so that's what I tell folks. Think of glycolytic, right? Glycolysis is predominantly an anaerobic process. Oxidative is going to involve aerobic. Okay? And we're talking about oxidative fibers, right? These are the muscle fibers that it takes longer to fatigue, so we nickname them fatigue resistant, right? And they undergo aerobic cellular respiration. So when you look at these following characteristics, it'll make sense to you. Think about it. If you're going to engage in aerobic cellular respiration, you need a lot of mitochondria, right? Because that's the cell organelle that engages in aerobic cellular respiration. Okay? Not only that, right? If you have a large supply of myoglobin, okay, that means we need to have an extensive blood supply to bring that oxygenated blood to the muscle tissue. Right? if we have lots of myoglobin, remember what I said I wanted to say last week, what I said last class, it's the myoglobin that gives muscle its red appearance. Okay, so the more myoglobin, the more red the muscle tissue will appear. But also because it has many capillaries. Okay, so this muscle tissue, right, this type of tissue has lots of ATP there, right, because we have lots of mitochondria, we've got a huge blood supply, and we got plenty of myoglobin, right? So our contractions can last for long periods of time. All right. Now, to contrast that, we have the glycolytic fibers, right? This is going to involve anaerobic cellular respiration through glycolysis. Right? If you think about it, you've got one glucose molecule who's only going to give you 2 ATP. You're not going to have a lot of energy, all right? And it won't last long for what energy you do get out of it. So these muscles are more likely to fatigue, okay? So if we have, all right, if we're undergoing glycolysis, we're not going to have a lot of mitochondria, so we'll have fewer mitochondria, all right? We have a relatively small amount of myoglobin available, all right, because again, we're undergoing anaerobic cellular respiration. We don't need oxygen. We have a smaller blood supply, so fewer capillaries, right? But we do have a large amount of glycogen. Again, we'll burn through that fast, okay? We can burn through that supply within 15 seconds, 10 to 15 seconds of intense workouts, okay? So these fibers, all right, will appear white because, remember, what gives the muscle fibers its red color, that's myoglobin, okay? So this does not have myoglobin, so these fibers will appear white. I had a picture. Here we go. Okay, so you can see, all right, the darker ones, those are going to be our oxidative fibers, and these lighter ones, those are going to be our glycolytic fibers. That's what we're talking about. So we have lots of myoglobin, these darker staining fibers, right? But we have no myoglobin and these lighter staining fibers. All right. So finally, all right, our glycolytic fibers, bless you, all right, they fatigue relatively quick. So those contractions last for a short period of time. Okay? Does that kind of make sense to you guys? All right. So how does a fast twitch fiber differ from a slow twitch? Okay? Fast twitch, they're going to have quicker contractions, stronger contractions, right? Again, everything about a fast twitch, all right, I hate to say this, it's going to be faster. Right? We're going to be able to propagate our action potential quicker. We're going to be able to release our calcium quicker. We're going to be able to reuptake our calcium faster. Everything about fast twitch muscle fibers, right, are going to be much quicker than our slow twitch. Right? So we can sustain a muscle contraction longer with fast twitch, right? But our contraction from slow twitch is going to be for a shorter period. Okay? Okay. How does an oxidative fiber differ from a glycolytic fiber? Pretty much everything I just described. All right. Oxidative undergoes aerobic cellular respiration. Right. They're fatigue resistant, which means it takes longer for them to fatigue. Glycolytic is going to be anaerobic. Okay. So they are going to lack our myoglobin. Right? They will have high glycogen stores, but unfortunately, we burn through that in about 10 to 15 seconds. So they tire quickly. Okay. So those are what we call the fatigable ones. All right, so the three types, now we're going to combine all of them, okay? So we got slow oxidative fibers, fast oxidative fibers, and fast glycolytic fibers. Okay, so those are our three types of skeletal muscle fibers. So if you look at it, slow oxidative, and these are slow twitch, okay? So these contractions will be slower, all right? Oxidative, okay, which means they're less fatigable. Okay, so it takes a while for them to fatigue, right? But because they're slow, they contain that slow ATPase. So these muscles have a high endurance. which is great because they're oxidative fibers, so they're going to undergo aerobic cellular respiration, okay? So we'll have a pretty decent amount of ATP supplied, right? So we'll have in these muscle fibers, we will have a decent amount of myoglobin there, so they will have some redness, all right, due to the myoglobin, right? And so these take a while for them to fatigue because they're slow fibers, but they're oxidative fibers, so they have a high amount of energy production from cellular respiration. All right, so our fast oxidative fibers, they'll have the fast variant ATPase, right? So that will be a quick, powerful contraction, right? But at the same time, we're lucky because they have the aerobic cellular respiration going on, okay? So again, we'll have that reddish color there, right? So these fibers you're going to see in numerous amounts in sprinters, okay? And then finally, our last are the fast glycolytic fibers, okay? They have the fast variant ATPase, fast, powerful muscle fibers, right? But they can't sustain their contractions for a long period of time because it burns through the ATP because of the glycolysis, that anaerobic cellular respiration, right? Because they're glycolytic, they don't have myoglobin, right? So they will have that white appearance to them. Any questions about that? Can I what? Yes. Keep it right there? Yeah, you got it.
I'm gonna go and type this slide up. I'm just gonna do it when I watch the recording again.
No, there's a lot of, trust me, when I was making this slide, this is detailed, not as detailed, but as condensed as I could make it, you know. I couldn't make it any less information on there. This is the bare bones.
Really?
Mm-hmm. Yeah. I mean, if you go into the section in the book and look at the slide, I managed all that onto one slide. And in the section in the book, it's, so you don't even, I shouldn't say you don't even need to read that section in the book, but this is the bare bones to it. All right. So where we're going to find some of these muscle fibers, all right, you're going to find certain muscles, all right, like I said, those muscle fibers, if you look here on our diagram, this is showing you the muscle, the single muscle cell, all right, muscle fiber here, and then you can see here are all the muscle fibrils made up of those different types of fiber types in here, okay? So, example here, all right? I love examples. In a hand, we'll see quite a bit of the fast glycolytic fibers, obviously for the quick contractions and the fine motor movements that we see in our hands. But, like I said before, our postural muscles, like in our back, are going to be made up of the slow oxidative fibers because they're more for endurance, right? They need to be able to resist fatiguing, right? So they need to be fatigue resistant, right? So, but they also need to have a significant supply of ATP. So they need to be oxidative fibers, plenty of mitochondria, plenty of myoglobin, lots, all right, so we can produce lots of ATP so we can do our kind of standing up, okay? Long distance runners are going to have that slow oxidative fibers there, all right, because they're going to be, they need to have the oxidative fibers predominantly for all that ATP production, okay? Sprinters, fast glycolytic, like I said, because they have to be able to generate that powerful, quick contraction, okay? Big thing here is this right here. Genes are going to be what primarily determine what type of muscle fiber is predominant in you. You can train and help modify that, but pretty much genetics plays a huge role. Okay? I don't have the genes to be an Olympic sprinter. Okay? I just don't have the muscle fibers for it. right? I've never seen a speed skater. You ever watch the speed skaters, how big their thighs are? Look at some of those Olympic sprinters, how big their thighs are. All right, I'm not asking you to look at my thighs, but my thighs, you could take 4 of my thighs and put them all together and equal 1 of their thighs. That's my point, all right? I just don't have the genes for that. And I could run and I could train until my feet fell off. It wouldn't matter. All right, skeletal, which muscle fiber type predominantly composes muscles that maintain posture? Slow oxidative. All right, this is a good point to stop and take a break, okay? And we'll take about a 10-15 minute break. And then we're going to come back. We're going to go over muscles just a little bit more, and then we're going to do some brain labeling, okay? So, like I said, I want to take a break here.